and I'm very happy that you are all here. Also, the people online, thank you for joining us. So there is one technical remark. Questions has to be put in a microphone because we have people online listening. And if you don't speak in microphone, your question will not be heard online. I'm Peter Niels. I am the chair of the Human Vaccine Committee at IABS. And I started this initiative, but I was very happy to have Kaat Bollards willing to join our group of scientists to put this up, and she did a marvelous job. I give the floor to Joris van der Putten, the past president of IABS.
Thank you, Peter. Dear friends, ladies, gentlemen, dear colleagues, it's a real pleasure for me as past president of IABS. I was president between 2016 and 2022. And I'm very happy to see that during this period, and even more now, that IABS is really continuing on its path that was in fact created in 1955 by Dr. Charles Mérieux. So probably you know or you don't know, the vision statement of IABS is access to health solutions for everyone, everywhere. And this access to health solutions, we do that since more than 65 years at IABS by bringing together all stakeholders. When IABS was created in 1955, you know, this was the period where the governments produced vaccines and biologicals for the people, and because the governments did that, by definition, this was excellent and good. However, there have been some problems on safety in particular, and this was the great idea of Charles Mérieux to say, well, we cannot continue like that. We have to sit around the table, at least to agree on pragmatic requirements for safety and later on immunogenicity and efficacy. So the way of working of IABS is always to bring together all stakeholders, so regulatory, governments, patients if needed, science, in order to openly discuss the challenges ahead or the problems that exist, in order to propose science-based solutions and applications, particularly on the regulatory side. So that's in this spirit that also these 2 days have been organized by Peter, Kaat, and many others, and I like to thank the scientific and the organizing committees. I like to thank warmly also the Secretariat, who did a marvelous job as always. And last but not least, I like to thank all of you to be present here, because this is just the objective that we are all present, either in face-to-face or those who are following by teleconference. So I like now to ask Jacob Kramer from CEPI to tell a word. You have seen the names of the sponsors, whom I really like to thank very warmly, and CEPI is one of our big sponsors. After Jacob, I will introduce Thomas, the next Thomas Verstraeten, who will also say a word. Jacob, the floor is yours.
Thank you. Some of you may have known Bob Small much longer than I did. For the past 4 years, he worked for CEPI and he was a member of the clinical development team. But he was also a member of the scientific advisory committee of this conference. We will definitely miss his expertise and experience, but also his dry sense of humor. Bob passed away while riding his bike on 3rd of June this year, and I'd like to invite you for a moment of silence. Thank you very much.
So now I introduce Thomas Verstraeten, who is the CEO and founder of P95, which all of you, I think, you know or at least heard about. Thomas.
Good morning, everybody. Thank you, Joris. So I've been asked to do two things. First of all, to introduce the objectives of this meeting, and secondly, to chair the next session. Now, there is a lot of interesting talks coming, and as usual, not so much time. So I will not spend too much time on the objectives, but try to save as much time as possible for the actual sessions. What are the objectives of this meeting? I think, and I kind of hope it's clear to everybody. We're here to talk about real-world evidence derived from real-world data to assist decision makers in both regulatory side and public health side with regards to vaccine development and vaccine deployment. To do that, we do two things. First of all, bring together people who have expertise in this field, right? We have a lot of people around the table, a lot of people online, excuse me, with great expertise in this field. So that, if you wish, is an objective by itself, to get those people around, to get those people to participate. And we're really excited to have everybody here. And I think just having the people be able to talk to each other and share experiences is already an objective met as we start. The next objective, of course, is to do something with that, right? With these discussions and with these debates, and to come with recommendations for how to do, if possible, a better job in the future. How concretely that will work, I guess, will be debated in the course of the conference, during the workshops, how to do this, how to communicate about this, how to bring this about. But that is certainly the other objective, is to come up with such recommendations. So I'll leave it at that. I hope it's pretty obvious to everybody, and I think in the course of these presentations that will become clearer and clearer as we hear about, you know, the experience of the different speakers that will be here over the next couple of days. So let me go to the next and first session, where we have the keynote sessions. Actually, we have 3 keynote speakers and 2 talks. Interestingly, the first 2 speakers have a combined talk, which is a duo talk, I guess we'll call it. So I'll introduce 2 speakers at the same time, and they will speak right after each other, and they have actually harmonized their talks, which is already a nice introduction to how we work together. So I will start with Helen Rees, who will be the first talker. I received the biosketch of Helen, and it's really a very impressive and lengthy biosketch. So if I go through the whole thing, I will shorten her talk too much, so I won't do that. So I had a challenging task to try and pick out the key parts. So Helen, excuse me if I miss anything critical. So Professor Helen Rees is the founder and executive director of WITS Research Health Institute in Johannesburg, South Africa. She's a professor in the Department of Obstetrics and Gynecology in the University of Witwatersrand. She's also an honorary professor in the Department of Clinical Research at the London School of Hygiene and Tropical Medicine, and an honorary fellow at the Murray Edwards College of Cambridge University. She's a medical professional by training who pretty much dedicated her career to improving public health in Africa. And pay attention to that last part because that's kind of a commonality with all the speakers that I will introduce. On top of all that, she's also the board chair of the South African Health Products Regulatory Authority. She has received multiple awards in several countries, including the UK, France, and South Africa. So that's Helen. Then we have Jacob. Dr. Jacob Kramer is a physician as well. He's specialized in internal medicine, tropical medicine, travel medicine, and infectious diseases. He currently heads the clinical development team at CIPI, and he's invited here in this position. Prior to working at CIPI, Jacob was the head of tropical medicine section at University Medical Center Hamburg-Eppendorf, as well as the head of the clinical research unit at the Bernard Nocht Institute for Tropical Medicine in Hamburg. In that position, he founded the clinical trials unit and was involved in several phase 1 to 3 vaccine trials, including research in Africa. Following this, he held position of a medical director, clinical development at Takeda's vaccine business unit in Zurich. And from there, he co-developed Takeda's norovirus vaccine, dengue vaccine, as well as polio vaccine. So without further ado, I'm inviting Helen to start with her presentation. Helen is not here. She will do this online. And Helen.
Thank you very much. I hope you can hear and see me a little bit, but thank you very much, colleagues. And I'm going to be talking today with Jacob about accelerated vaccine development pandemic situations and how we can use real-world data and real-world evidence together with clinical trials. Next slide, please. So starting just with a few slides about what we understand as real-world data. Well, real-world data is derived from many sources and often comes from heterogeneous patient populations in real-world settings. Tends to be observational, is often collected in clinical practice, but also in real-world environments. It can be used to evaluate effectiveness and safety of treatments, products, and patient programs, so it's diverse in what it can be used for. It provides more comprehensive data compared to clinical data, but it can be used to complement evidence from clinical trials, which is the focus of what we're going to be talking about. Next slide. Here are some examples of real-world data sources. Electronic or paper-based patient records, which will have demographics, medical procedures, patient and disease registry. We're all familiar with cancer registries, but big programs like the PEPFAR program with HIV and TB. Patient-reported outcomes, information on symptoms, and very often this can be patients reporting adverse events in relation to a vaccine or to a medicine. Administrative claims and billing, this can be hospital records, emergency admissions, or in some settings, insurance company and public sector listings. Social media platforms can also be used and are increasingly important to answer certain questions from broader community feedback. And in more sophisticated settings, wearable devices, for example, with insurance companies, are also increasingly being used. Next slide. But there are challenges associated with using real-world data. The data quality can be inconsistent, inaccurate, or incomplete. And particularly, there can be errors or biases, who's reporting and who's reporting on who and why. Very importantly, and Jacob will talk more about this, what is the comparator when you're looking at real-world data? Something that we've experienced here looking at large data sets is the lack of standardization in data collection, definitions, coding, different regional interpretations. And then also increasingly important questions about ethics and the privacy of sharing this data, particularly when with regards to patients. You can have huge numbers of data sets. There's lots of data out there, but it can be difficult to get access with all of the above points. And then you have to think about how do we coordinate public and private, because if you don't do that, say at a national setting, biases will come in. Next slide, please. So clinical trials and real-world data can, however, be complementary, and this is what we want to explore, particularly in an emergency setting. You're all familiar with clinical trials. They are controlled, they have comparators, strict inclusion, exclusion criteria, strict monitoring. There's informed consent. and increasingly community engagement is seen as very important. But real-world data can be used to complement clinical trials. It can help to validate findings from clinical trials and inform clinical trial design subsequently. And because we're talking regulatory, it can support regulatory approvals and post-approval validation and safety of healthcare products. It can also be used to identify unmet healthcare needs and particular patient subgroups that are not included in clinical trials. And for this reason, real-world data can be used to complement clinical trials in emergencies. Next slide. Are we stuck on the slides? No. Oops, we got excited there. Okay, so, but we're also talking about real-world evidence. Are they the same thing? But real-world data is what is used to produce real-world evidence. So they are closely related, but they're not the same thing. We use real-world data increasingly with machine learning, AI methodologies to actually try and understand what the data is telling us. But real-world evidence then uses that data to interpret what is going on, to look for patterns and to look for recommendations. Healthcare stakeholders are increasingly using real-world data to help make decisions around policies around healthcare services and the rollout of therapeutics and vaccines. Next slide, please. So I'm going to cover now 4 examples of the use of real-world data in somewhat different ways that complement clinical trials: COVID-19, Mpox, yellow fever, and Ebola. Next slide. So COVID-19 you're all familiar with, and here we used real-world evidence quite extensively, but it was based initially on data from clinical trials. We had large randomized trials. These were only used, the enrollments were only in seronegatives. We had endpoints which were frequently mild to moderate COVID-19 disease. Trial participants had to be unvaccinated, and so it was a rather healthy trial population. The vaccine efficacy could only be assessed after a few months, and at the time when we did those trials, we had limited numbers of variants. So to look broader and more broadly, what did we then do? We looked at real-world evidence generated from rollouts. We looked at different subpopulations and age groups. We looked over a longer period of protection. We were able then to look at the interplay with products, and particularly vaccines with variants. And we looked at protection against less frequent events such as death. Next slide. So this is the first example. I'm going to give 2 examples of how we use real-world evidence. And this is from routing data from the Danish data set, which looked at vaccine effectiveness and hospitalization with 3 variants. And this was a nationwide Danish cohort study. Please press again. So here, the Danish nationwide cohort study collected data on populations over 12 years old. They estimated the vaccine effectiveness against infection and against hospitalization with those variants. And they looked at 2 doses, and they found that it provided high protection against SARS-CoV-2 infection and against hospitalization with the Alpha and Delta variants. The 2 vaccines provided, however, only limited and short-lived protection against Omicron. But the protection against hospitalization was higher than against infection. And a 3rd dose substantially increased the level of protection. So this is one example from a national data set. Next slide, please. You can click again. The second example is from the Israeli data. Similarly, the Clalit Health Services has a large data set looking at healthcare coverage with over 1 million people who were eligible to be included in this study. If you look at the graph below with those very large numbers, they were able to look at what happens with 2 doses in terms of cumulative incidence of infection and death versus 3 doses. And you can see that from the blue line at the bottom, that 3 doses were more effective at protecting against death than 2 doses. This again is using large data sets to give an interpretation that you could not get from a clinical trial. And complementary to this, they were able to also look at safety. Next slide. Mpox is more recent, and as you well know, over 100 countries experienced this unexpected outbreak of Mpox, different to those that we had previously seen in the West and Central African region. So from this German study, when this occurred, there were 3 vaccines that were potentially eligible for use that have shown some sort of effectiveness in clinical trials against Mpox. And this was evaluated, the MVA-BN vaccine was evaluated. This looked at 21,000, when this started, there were 21,000 cases worldwide, and Germany had got about 2,500 confirmed cases. And so they started a vaccine strategy with immediate vaccination versus delayed vaccination with the MVA-BN vaccine. And they noted that as the vaccine at that stage was only available in limited quantities, that they recommended as a result of this study that post-exposure prophylaxis should be offered to exposed individuals as a matter of priority. But in addition, individuals with an increased risk of a severe disease, for example, those with immunodeficiency, should be vaccinated with priority. So there were 2 recommendations that came out of this real-world data, which was those who were at increased risk of severe disease as a prophylaxis and those who had been exposed. Next slide. The second Mpox study we're going to look at was undertaken looking again at the MVA vaccine, and this was done in France. Sorry, this was done in France. Yes. In this case, the main objective was post-exposure prophylaxis failure after 1 dose, with positive Mpox PCR being the indicator at 28 days post-vaccination. They had a small number of participants, but again, this kind of interpretation of the vaccine had not been looked at before. Here they used a ring vaccination strategy, looking at people who were exposed to an individual who was confirmed to have had Mpox. And again, they were able to show that this strategy looked to be effective, but the results are still coming out. Next slide, please. Just going back in time, and something that is a little bit different, but again, looking at real world data, is looking at real world evidence for yellow fever. In this case, in 2016, there was a big outbreak of yellow fever in Angola and in the DRC. There were stockpiles of yellow fever vaccines, but insufficient for the numbers that were then required to be vaccinated. In particular, there was a huge concern about an outbreak in Kinshasa in the DRC, where up to 10.5 million people were at risk, but only 5.8 million doses were available. There was one study that had been done in Brazil with 1 vaccine, with 900 men, which had looked at de-escalating doses of that vaccine with 6 phases of de-escalation, which looked like it was both safe and immunogenic. So on the basis of that one study, there was a rollout of fractional doses in Kinshasa, using 1/5 of the standard dose. There wasn't a control group, but there was follow-up looking at safety, immunogenicity, and the impact was very significant in terms of preventing this outbreak spreading. Next slide, please. As a result of this, and one of the things that we know about real-world data and real-world evidence, I'm just giving you this as a highlight, is, as I said at the beginning, real-world evidence and data can inform a research agenda. And this was the research agenda on fractional dose yellow fever vaccine that WHO published in 2017. They asked the question, are all WHO pre-qualified vaccines suitable for fractional dose vaccination? Similar questions being asked now about HPV vaccination. You can't assume if it works for one vaccine, it will work for all. They asked about different age groups and particularly about children using fractional dose. The questions asked were about immunogenicity and vaccine effectiveness, but over a longer time period, 5 or more years after vaccination. The question was asked about whether you would get the same effect against different environmental exposures and different epidemiological and genetic backgrounds. The other question, very relevant in Angola and in the DRC, is what about people with asymptomatic HIV infection? Will fractional dose work? And then finally, do you get fewer severe adverse events after vaccination with fractional dose? This was the agenda that was generated from the real world data that came out of that fractional dose introduction. Next slide. And then finally looking at the Ebola vaccine, here again, as you remember, in West Africa, there was a huge Ebola outbreak between 2013 and 2016. And once again, there was a vaccine that was thought to be effective from challenge models and with some phase 1 and phase 2 studies. This was the RVSV-ZBOF vaccine. You had to get into the field really rapidly. Many designs of studies were proposed, but based on this very limited data, the study that actually did show effectiveness was a novel cluster randomized control trial where people who were contacts or contacts of contacts were given the vaccine either immediately or with a delay of 21 days. And it was found that those who were offered the vaccine immediately had 100% protection, unlike the cohort who had the 21-day delay were 16 cases. On the basis of this vaccine has now been widely used in Ebola rollouts. But again, the secondary research agenda, looking at long-term protection and all the other questions that we've seen there, pregnant women, younger age groups, also was assessed. Next slide. So just a thought on this, which is again something we should think about when thinking about how we use real-world data and evidence. What are the ethical considerations about using or generating real-world data, rather, in an emergency versus in a controlled clinical trial? Well, the first thing is specification of exceptional circumstance. The West African Ebola epidemic was seen as an exceptional circumstance, but in the future, what is an exceptional circumstance? A pandemic certainly is. What about more localized outbreaks, Mpox being a case in point? How do we define exceptional circumstance when we will use this kind of data? Secondly, what are the specifications for an unproven intervention? What is enough evidence to allow us to use, for example, a vaccine? What do we need to know about efficacy and toxicity? Is animal data sufficient if you've got a condition with high mortality? What is the risk-benefit ratio, and how do you weigh that up? And the third question is the goals of the interventional research. What are we trying to do here? Are we trying to protect individuals in an emergency? Or are we trying to do a larger effectiveness and safety understanding? So what, from an ethics point of view, are we trying to achieve? Is it the individual benefit or is it the public health benefit? Next slide, please. So this is the conclusion of this part of the presentation. I'm going to hand over to Jacob, but in conclusion from here, real-world evidence can give you excellent evidence and real-world data can be excellent, but there are limitations. You need to have a functioning public health system, which is key if we're going to use real-world data. There are real concerns about data protection and data sharing and privacy. Timing is key. We know from those recent Ebola outbreak in Uganda that even with 45 days to get into the field with a vaccine, we didn't get there quickly enough. And again, a vaccine with limited data. Importantly, what are the comparators? And then how does what we generate contribute to a new research agenda? Thank you. I'm going to hand over now to Jacob, who will take this discussion further. Thank you very much.
Thank you very much, Helen, and I will build on what Helen just presented and shift to the question, what does it need in pandemic situations? Or let's be maybe not that ambitious and always think about pandemics, but lower level as well, so epidemic conditions. Next slide, please. Or can I actually move with this marker? Yeah, great. Okay. So you already saw some definitions, but I'd like to place this in context in terms of pandemic preparedness. And I think we have to stop thinking in conventional manners when it comes to emergency situations and pandemic preparedness. So is real-world evidence always what we refer to as a phase 4 trial? And if yes, what is a phase 4 trial? And if you look here in the cancer research, for example, it's to generate evidence after a drug, in this case a vaccine, has been licensed. So phase 4 trial, based on all the evidence that has been generated in support of vaccine licensure, is to generate more evidence about side effects, including rarer side effects, long-term risks and benefits, and also how the drug works when it's used more widely. So is this the case in pandemic situations? Maybe the vaccine is not licensed, at least not through a conventional pathway. Maybe there's some sort of emergency use authorization in place and lack of evidence. So if we think about moving from maybe prototype-specific data, phase 1, phase 2 data, directly into phase 4, real-world evidence, that will not work in every outbreak. And as said, I think we have to think in different terms. And I'll come to this and explain what I mean by this through the next slides. So evidence generation, you need information on vaccine efficacy and vaccine safety. And we can generate that evidence based on controlled clinical trials or real-world evidence. And sometimes the term RWE is even confusing, and maybe this is something we can align on after this meeting, that RWE is actually real-world evidence, and not just limited to real-world effectiveness, as it is also sometimes referred to. So we have to generate data on efficacy or effectiveness. But the point I'd like to make is that in clinical trials, you cannot generate evidence on vaccine safety in a manner that it is really complete and gives you a full picture. Every pre-licensure vaccine development program will be too small to detect rare safety events. Of course, you always do, the trials are always called safety, immunogenicity, and maybe efficacy trials, and you have a chance to detect safety events up to a certain frequency. But this is really only good enough to rule out maybe that a vaccine is completely unsafe. So if you see frequent safety events, you would probably not even go further. But the rare events, and think of the COVID-19 vaccines with myocarditis and the thrombotic events, TTS, they only popped up from using the vaccines in large quantities. So for this, you can only rely on real-world evidence. So what do we do in pandemic situations and outbreak situations? Is it controlled clinical trials? Is it real-world evidence or something in between? I think there's two areas that define what type of evidence we can actually generate. One is the hard facts, if you will. So what is the pathogen? What is the route of transmission? What is the population at risk? All of this, of course, feed into the trial design and what type of evidence generation strategy you would select. What is the size of an outbreak? What is the dynamics? How long does it last? And also, so the timing is important, but also whether or not this has to be seen in context of other, maybe non-pharmaceutical interventions. So this is really informing the study design. Can you do a large trial? Do you see enough cases to generate statistically significant information? But at least as important, I think this is really where the gray area lies, is the second aspect, the benefit-risk ratio. So what is the risk appetite of the regulators, of the population, of the politics to use a vaccine with, I shouldn't say limited data, and you will see that we always need data, but maybe going ahead and right away using a vaccine and generating vaccine or evidence on its efficacy while actually using it. So this, of course, depends on the case fatality rate. Just think of something that we may not know a whole lot about, but the case fatality rate is 50%. And as we speak here, there's another 3 people that die outside on the sidewalk. The benefit risk assessment will be completely different as if we talk about a respiratory disease. And I don't want to underestimate the public health relevance of COVID, but of course, it's not, it's much lower than 70% case fatality rate as we see for other diseases. What do we already know about similar vaccines, potentially? Can we leverage on existing evidence? What do we know about the vaccine platform? Is there a vaccine license based on that same platform that we can, refer to? And so on, and certainly also what are the perspectives of different regulatory authorities, and I'm sure the perspective will be different on an Ebola outbreak somewhere or a Nipah outbreak, depending on where the regulator actually sits. So let me deep dive a little bit into these two areas, starting with the outbreak dynamics. And I think it is clear that there's a lot of areas that influence the design of a clinical trial and whether or not real-world evidence or clinical trial can be done or neither of the two. And this depends on the outbreak size, the speed of transmission or not, the duration of the trial. Sometimes they're short-lived and you have no chance to even start any type of evidence generation. The level of pre-existing evidence, will you right away move into real-world evidence, not do a controlled clinical trial? The quality of the source data, real-world evidence, of course, relies, as we heard from Helen, on the availability of good data, trash in, trash out. So if you do not have a functioning public health system where these data are collected prospectively, or if you do have a public health system, like in Germany, but people don't like to share their data, so data protection issues, real-world evidence, actually, like we've seen the data from Israel and from Denmark, will become difficult. So in the end, it's not either or. It's not clinical trial or real-world evidence. And I think it's a no-brainer to say that both real-world evidence and controlled clinical trials can rapidly generate evidence when the incidence is very high. If we run to really a huge outbreak, just in terms of hard facts to do clinical research. And neither real-world evidence or clinical trials can rapidly generate evidence if an outbreak is really very small. Think of Nipah. We have an outbreak right now in Kerala in India. There's less than 700 cases in the disease history. Usually an outbreak is 5 cases, 7 cases, 12 cases. That's not enough for any type of evidence generation. Time. And just to illustrate this with an example, so this is kind of a map of the city of London, just about 9.6 million inhabitants. And think of a scenario on the left-hand side where you have just 400 cases scattered across the city of London, small clusters, and it takes really 6 months realistically in public health system to vaccinate 50% of those at risk. So it's really a very defined risk population. It takes time to roll out a vaccine and to convince people to get vaccinated, to get data into public health records that you can then use to analyze in terms of real-world evidence. On the other end, 400,000 cases in 12 weeks, rapid human-to-human spread, so probably respiratory disease. And again, it takes 6 months to vaccinate 50% of the entire population this time of London. And we have examples, so think of monkeypox, for example, on the left-hand side, think of COVID or something similar, respiratory disease on the right-hand side. It takes time until you roll out vaccine. So neither a clinical trial nor real-world evidence is probably generating evidence. I mean, we think of 100 days, and I'll refer to this, come to this, the second part of this. But it takes more than 100 days to generate this type of evidence. And how comfortable, again, back to my benefit risk assessment, how comfortable are we to work with vaccines without having this confirmative evidence? So the second area is much more difficult, of course, and the benefit risk assessment, what is your risk appetite and what are the consequences? And just to go through a few examples, and I'm not saying that licensure is benefit and epi is risk, but just to illustrate a little bit what I mean by this with different pathogens. In 100 days, what we are talking about in CEPI, we do this already for decades in the influenza world. We don't generate any additional clinical data while adopting the seasonal influenza vaccine to the seasonal strains. So this is something that we do with a lot of comfort, I guess, because we have tons of data and we have decades of real-world evidence available, and we know the platform technologies, and we know they're not really a safety risk. Monkeypox, it's difficult. Yeah, there's a code of protection. That's what current vaccines have been licensed on. There's some animal challenge data. But the epidemiology is really challenging, and the case fatality rate is low. So this is a completely different benefit-risk ratio. And the epidemiology, of course, is different in endemic areas in Africa as well as compared to some populations at risk recently in high-income countries. Chikungunya, about to license vaccines, again, based on code of protection and animal challenge data. But the epidemiology, we have seen very small outbreaks, just in a province in Italy. We have seen country-wide outbreaks in Paraguay, for example, neighboring countries. And we have seen gigantic outbreaks all around the Indian Ocean, affecting hundreds of millions of people with chikungunya. We just don't know when and where the next outbreak is going to happen, so how to generate and prepare for respective evidence generation. Ebola, Zaire, we can predict there will be a next outbreak. There will be a next outbreak somewhere. They have been small, mostly at least, with exceptions, of course, and they have a very high case fatality rate. We have a licensed vaccine. Are we now willing to license the Sudan vaccine without further vaccine efficacy evidence, simply because we would say, well, what's the difference? Sudan, Zaire, Ebola virus vaccines. And then Nipah already mentioned there's less than 700 cases in the disease history, so we can do neither real-world evidence nor a clinical trial, at least with the existing epidemiology. But what is disease X in the end? So what do we prepare for? And in the end, it's probably one of the four levels. It could be just a strain change. Think of COVID, and probably, at least with some of the platform technologies, we are there now to just adopt new variants without generating a lot of efficacy evidence, at least from clinical trials. It could be an outbreak with something known. We know the virus. We've never seen really a very, very large outbreak, and we do not have a vaccine, at least not in advanced stage clinical development, for example, Nipah. So it's not really a disease X, it's an outbreak X, if you will. Could be a novel pathogen of something that we already know, so new poliovirus, for example, new enterovirus, or it could be a completely novel pathogen that's unrelated to something that we have already seen. So we have no data that we can leverage on, at least not from the pathogen. Maybe we have some platform-specific data, but that's really unlikely. I think the disease X will be a variation of something that we already know. And then we can always leverage on existing data. I think this is an important consideration here, and this is something we discussed extensively during the COVID-19 pandemic in the so-called clinical development SWOT team. How long can you actually do a randomized placebo-controlled trial? And it's a no-brainer that you can, of course, do a randomized placebo-controlled trial when the vaccine is available but not licensed. But you can still do a randomized placebo-controlled trial if a vaccine is available and licensed, as long as it is not yet recommended for use, kind of the standard of care. So there's no NITAC recommendation available yet. And even if there is a NITAC recommendation, so there's a vaccine available, it is licensed, and you have a vaccine use recommendation in a specific country, you could still do a clinical trial if the vaccine is not available in that country, and you can make the vaccine available through a clinical trial. The ethical questions, have to be seen in parallel, and there's a lot of debate, and you know that randomized placebo-controlled trials have still been done with COVID vaccines while they were recommended and available, but not available in each and every country. So in principle, what you can for sure say is if all these four boxes are ticked, it is difficult to do a placebo-controlled clinical trial, unethical. So then you will have to rely on real-world evidence. So what is needed? Over the last few slides, I'd like to kind of lean over to the CEPI 100-day mission and kind of see this, what Helen and I discussed so far in this context. And some of you may have seen this slide before. This is the conventional vaccine development timeline. Takes years. You do sequential phase 1, 2, 3, licensure, and then you start with real-world evidence phase 4, and so on. In COVID-19, we have seen in accelerated timelines, still sequential clinical trials, but rolling reviews, regulatory agency reviewed data in parallel, so we were able to speed this up. We in CEPI did an analysis. Could we accelerate this even further? So we took all vaccine programs and took fastest clinical trials, fastest activities, and kind of combined them, and we landed somewhere around 250 days. So based on conventional vaccine development, you cannot shorten vaccine development timelines further or lower than 250 days. So why 100 days? And 100 days really requires a paradigm shift, if you will. And to illustrate this a little bit in a larger slide, it relies on what you do before day one. So you will go through the vaccine families and you look at pre-existing data and so on. You generate a lot of phase 1, phase 2 data. You learn much about the platform technologies, and maybe you can license a vaccine based on that platform for other pathogens. And we know the 25 viral families. Some disease X will always come out of one of the existing viral families. Within 100 days, you really just adopt your vaccine to the outbreak pathogen and you describe the outbreak. You need to learn about the benefit-risk assessment. How dangerous is that outbreak actually? Is there human-to-human transmission? What is the case fatality rate? What is the population at risk? And then maybe based on all you have, how dangerous is the disease? How much pre-existing evidence on prototype vaccines do I have to license or authorize, emergency authorize a vaccine? And then what do you do then? To confirm the evidence on the actual outbreak vaccine. Is it a clinical trial? Is it real-world evidence? Is it something in between? And I think it's really both. And I would like to illustrate this a little bit with this slide here, or actually this one here. So you do a lot of work based on prototype vaccine, dose selection, first safety information and so on, and then you park your vaccine. You have your prototype vaccine, and as soon as you run in an outbreak, you have to, within the 100 days, characterize the epidemiology of the outbreak to really understand the risk and the threat actually of this outbreak. And then just to illustrate this, and I'll go through some examples on the next slide, a randomized control trial, real-world evidence, manufacturing at large scale, and further review has to happen in parallel. But there's no one strategy that would fit all types of outbreaks. So if you look at this separately for different pathogens, and this is an example for the COVID-19-like pandemic, if you will, for example, SARS-3, so you have a vaccine, that's what CEPI is currently working on, prototype vaccines for future coronavirus, for example. You describe the outbreak, and what has been done in the COVID pandemic is randomized controlled trials generated evidence within 350 days on vaccine efficacy, and they were followed by real-world evidence. So it was really both that generated, and Helen has shown this in her example. But there might be examples where clinical trials are not necessary, and I already brought the example of the influenza pandemic. So just real-world evidence is the source of generating evidence in support of vaccine use. There might be, and this is by no means discussed, this is just for illustrative purposes here, there might be other examples where real-world evidence is actually difficult. For example, maybe Ebola, you run into a small outbreak, you do these early versus delayed ring vaccination trials, and maybe the outbreak is over before you actually generate enough evidence. So the actual licensure to make vaccine available could happen even based on what we know about Zaire, and you go ahead right away to approve a Sudan virus. Or you do an early versus delayed ring vaccination trial and approve it then. Maybe the outbreak is over before you have enough evidence and you have to repeat this. And eventually, over a series of outbreaks, you will generate that evidence, but you're still confronted with a disease of case fatality rate of 60%, and you do not have a licensed vaccine. So this is the benefit risk assessment that has to be made in the context of the selection of your method for evidence generation. So if we do this, for example, if we look into pathogen, work on our priority pathogen, we have this list, the WHO blueprint list of priority pathogens, and we work on vaccines, and we have to think of strategies to generate evidence on vaccine efficacy. Based on the current outbreaks, Nipah, it's very difficult. So maybe we can start, and there was someone, Stephen Luby from California, he said in a conference it would take 516 years to recruit into a conventional controlled vaccine efficacy trial. So that's not possible. But maybe the outbreak changes, and we also need to prepare for this, and this is exactly what happened with Ebola. I mean, Ebola before 2014 was like this. A few cases here, a few cases there, and then all of a sudden we saw this outbreak in West Africa. This is something that could happen with Nipah as well. It occurs in India and Bangladesh and Malaysia and highly populated areas. So we need to prepare, and we have a whole toolbox of evidence generation techniques, be it clinical trials, early versus delayed ring vaccination trials, test negative designs. I'm sure we'll discuss more in this conference that we need to prepare for. If we go through all of our priority pathogens, Nipah, Rift Valley fever, chikungunya, we work on coronavirus, different scenarios, Marburg and so on, we will eventually approach our disease X as well. I think if we have gone through, and there's mosquito-borne diseases, there's human-to-human transmission, there's respiratory diseases in here. So we have pretty much all scenarios and all outbreak scenarios, and there's more scenarios behind each priority pathogen. I said, we have seen chikungunya in the Emilia-Romagna, we have seen it in Paraguay, we have seen it around the Indian Ocean. So there's within the chikungunya box here already several different outbreak scenarios. And if we prepare with evidence generation strategies, using the toolbox we have, real-world evidence, controlled clinical trials, something in between that we still have to invent, maybe, probably approach disease X as well. So to conclude, time is key. How quickly do we need to generate the evidence? the control group, and Helen mentioned that as well, in the end, the control group is the problem. I mean, you cannot really generate evidence if you don't compare something. And is it ethical in some situations to have to randomize someone into a control group, so no intervention? And that actually applies to both real-world evidence and clinical trials. We need scenario-specific approaches, and we also have to define, in the context of real-world evidence, is it a classical phase 4 trial? What do we really mean by vaccine approval? Is it really full licensure? Is it kind of some sort of emergency use authorization? What is the appetite of the regulators to actually allow the vaccine use without, seeing complete data and maybe generating evidence on vaccine use while actually using the vaccine? So phase 4-like, trials are done after a drug has been shown to work and been licensed. That's not the case, or not the case in many outbreaks that we will face. And in the end, there is no blueprint strategy. It's not real-world evidence or clinical trials. It's probably both or something in between. Thank you.
Do you mind staying for? Yeah, so thanks very much, Helen and Jacob, for very nice presentations. You went a little bit over your time, but that's fine because we were quick to start off. So we have about let's say 5 to 10 minutes for some questions. The way we do this, so we ask people online to write their questions, and Peter is gathering those questions. So I'm trying to find Peter there a little bit in the back to see. Peter, do you have questions from... Sorry? Not for the time being. Okay, so let's go here and see if anybody has a question here in the audience. Yes, Steve, go ahead. Yes, Steve, you can. Oh yeah, as you wish.
Anyway, thank you both for a great presentation. I think that my comment is, I think the dichotomy between real-world evidence and RCTs at that boundary, I think we need to explore that more because real-world, phase 3 trials that I've done for licensure have used real-world evidence, and I think that if we dichotomize it, we lose something. So I'd sort of like to get your comments on that as to whether we can innovatively explore how to best harness the best of both worlds, if you will, in future trial designs.
Helen, Jacob, let's see how coordinated you are in agreeing who will answer the questions.
So I can start and then hand to Jacob, and then we can do reverse next time. I think that's a very good point, and this is where I think risk-benefit also comes in. It's what is your tolerance for moving very quickly from one scenario to another? And we gave examples of where there's very limited clinical data, but we had rollout. But probably what we should do is spend a little bit more time seeing if there are some very concrete lessons learned from not only the examples that Jacob and I gave you, but there are many examples where we are sort of interfacing clinical trials and real world evidence. But in terms of speed, you're absolutely right. We need to work out how we do this more seamlessly and under what circumstances can we move very rapidly.
I'm not even sure, Steve, if it was a question or a statement. I can only agree if it was a statement. But maybe just to add, it's to what Helen has already responded. It's not just a question of the specific scenario. It's also a question of the perspective and the regulatory agency. And I think we should move away from just assessing this with stringent regulatory agencies, as they're not affected with many of these outbreak pathogens. And it's important to enable regulatory authorities in the area where these outbreaks happen to come up with their own benefit risk assessment and maybe strategies. But to think of innovative trial designs and maybe even something in between, kind of to lift the boundaries of conventional vaccine development, that's really a moonshot in itself, I would say. And so really appreciate, for example, this conference to look into this, and hopefully more to come.
I wonder if I could just add on the regulatory agencies, because just a small comment here. We're moving away from the language of stringent regulatory agencies. For those who are not familiar, WHO is now looking towards giving maturity levels and having a WHO-listed authorities. And many of the African regulatory authorities are now seeking, you can go from 1 to 4, 4 being the highest. Many of them are seeking a maturity level 3. And in that, you can apply for a maturity level separately for vaccines, separately for medicines. I think that one of the things for this conference should be to think about how do we develop that dialogue with regulatory authorities, because it's quite novel what we are talking about here. And it's not necessarily going to be something that regulatory authorities in affected countries will have given much thought to. So I would support an interface after this with regulatory authorities.
I guess my point was, I think it's not good if you go to regulatory agencies and say, well, this is only real-world evidence. It doesn't meet the gold standard of a phase 3 randomized trial. Many phase 3 randomized trials, because of the select populations, have yielded misleading results. That's true for pneumococcal conjugate. It was true for rotavirus effectiveness, et cetera. So I think if we could describe the fact that by playing to the strengths of both and coming up with a hybrid approach, we would be better off. I think the regulatory agencies might be more willing to accept real-world evidence as good evidence for licensure for a trial if they realize that the RCTs have been set up as the gold standards, but there are a lot of examples where the results have not really been consistent with what's happened when the vaccine has been introduced.
All right, thanks. I saw a few more hands, maybe one more question. Hector, do you want to go ahead?
Yeah, I was curious, you both mentioned
Could you please introduce yourself, especially for the people online?
My name is Hector Izurieta. I work for the FDA, Food and Drug Administration. And I'm curious about the example you presented about the Ebola 2012-15 outbreak in West Africa with thousands and thousands of deaths. The ring vaccination trial was successful, but ended and provided data when the outbreak was basically over. Could you elaborate? You both commented about that example, and I found it interesting because it's relevant to this meeting. Would you like to further comment on that specific example?
Thanks. I can start, Jacob, if you want.
Yes.
So the ring vaccination strategy was adopted from the smallpox strategy. At the time, and I was very involved with this, there were many different clinical trial designs on the table. The only one that got into the field fast enough to generate a result was the ring vaccination strategy of the RVSV. So that was sufficient to give confidence that, and it's a single dose, I should have added that. That was sufficient to give confidence that this was an appropriate vaccine to use. But as we said, because of the very limited conditions, there were sufficient cases. As I say, in the delayed controlled group of 21 days, there were 16 cases. And this was statistically significant because these were large clusters of contacts and contacts of contacts that were randomized in that manner. That there has been follow-up of the people who received the vaccine, obviously looking at longer-term protection, immunogenicity, also looking at, do you need to give boosters? There's also follow-up and interest because of whether or not to be offering the vaccine to healthcare workers. But that was a conclusive finding. But, as you said, it's one of the challenges we have. How do you design trials for outbreaks? One of the other things with regulatory authorities in affected countries is to actually do trial designs that are approved both by regulators and ethics committees, so that in the event of an outbreak, you can move very rapidly into the field. And this is also being looked at by CEPI, by Gavi and others in terms of having stockpiles that can be rapidly mobilized. But in fact, in my opinion, that was a clinical trial that showed in those extraordinary circumstances and efficacy of the vaccine and safety.
Jacob?
Yeah, and just to add from CEPI's perspective, but this applies to many other organizations, whenever you run into an emergency situation, you get really very nervous and you don't waste the crisis, let's generate evidence, let's do something. But I think we have to be patient as well. I mean, yes, we need to do something and we need to prepare for this outbreak, but the evidence that we generate, even if it's incomplete, will always be good for something. There will be a next outbreak, there will be a similar pathogen. The only way to really establish vaccine efficacy, the only theoretic way to establish vaccine efficacy in peacetime, if you allow this term, is based on quality protection, if we have one. And then you would need to do real-world evidence to confirm vaccine efficacy while using it. But that is a concept, it's only a theoretic way forward. It doesn't work out in many pathogens. The regulators want something measurable, reproducible in terms of immune response, and that is difficult. Sometimes we don't have animal challenge models, so we do rely on clinical evidence, be it controlled trials, be it real-world evidence. Single outbreaks may not be and will not be in future enough or long enough to generate that evidence. And I think we also need to be a little bit innovative, of course, but also patient. We have generated a lot of evidence that in the end wasn't really helpful. Thank you.
So thank you very much, Helen and Jacob. I think that was a great opening and already quite a lot of material for discussion, and we've just touched on a few of the topics. So, but before we run late, I'd like to introduce somebody who is, I don't know if I have to say good evening or good morning to you, Brad, because you're almost on the other side of the world, right in Alaska. So thank you very much for still being awake and being here with us. So Brad Gessner is Vice President, Global Medical Lead for pneumococcal and Lyme vaccines at Pfizer. Brad is also an adjunct professor at the Center for Vaccine Development of the University of Maryland. Prior to his position at Pfizer, Brad was the unit manager of the MCH Epidemiology Unit for the state of Alaska. And Brad has always been associated to Alaska, as we know. But he's also been the director of the Agence de Médecine Préventive for Africa. And there is our link with the other speakers again. So Brad, please, the floor is yours.
Okay. Can you see my screen?
Not yet. Just give us a second. We're still seeing the beautiful town hall of Leuven.
Says I'm sharing. I mean, I've sent it to Maranita in case it's not working.
Yes, we can now see it, Brad. Go ahead.
Okay. Let me go into presenter view. All right. So you should be seeing the full screen, right?
Actually, we're not. We're seeing your kind of screen with 2 slides.
Okay. Well, I don't really need it. I'll just put it into the normal presenter mode and then we'll get going. Okay.
All right, that's great.
Does that look good? Okay.
Yes.
So this will be, rather than the outbreak type of vaccines that you heard about before, this is a talk more on PCVs and RSVs. And by the way, I should make a correction. I did do PCVs and Lyme vaccines. And then now I am the global medical lead for respiratory vaccines and antivirals. So that's PCVs, RSV, and RSV antiviral. I no longer do Lyme disease. I am an employee of Pfizer. All these views are my own, not necessarily Pfizer's. So the first thing I'll just mention is this, and I guess the other big difference is this is more a perspective from more developed settings, so Europe, North America type of settings. And this is a document that the FDA's put out, considerations for the use of real-world data and real-world evidence to support regulatory decision-making. I think this is a really fantastic document, and it just shows how far people have come in thinking about these in some of the regulatory agencies. This is just a little background of real-world evidence for the purposes of this presentation. So I think this has been defined before, what I mean when I'm talking about real world evidence or data, and that's registries, electronic health records, medical claims. I think Helen went through a lot of other sources that potentially could be used. I'm not talking about prospective observational studies, although I will give a sort of a counter example of how that's being used. So only non-interventional observational studies does not preclude the collection of additional data. So this is specifically through things like database linkages, lab and radiology results, I think are really critical, or questionnaires. It can be considered for vaccine technical committees, also known as NITAGs, or regulatory agencies. As was mentioned before, safety outcomes are critical and really are outcomes that are mainly derived from real world evidence, but that's a whole topic in itself, so I won't discuss that. And I don't plan, since I only have 15 minutes, to say much about design and statistical methodology, and specifically things like a randomized versus a non-randomized vaccine allocation. So, for example, the FDA, in some of their original guidance a few years ago, it was pretty clear that the goal was a randomized allocation and then using real world evidence databases to look at the outcomes. And then I'm also not going to discuss approaches for the observational designs. But what I am going to discuss are two things. So one is exposure sources and definitions. And I should say this is all gleaned from, the last 5 or 6 years working with Pfizer and trying to use real world data and really trying to push the envelope on what could be used for regulatory purposes. So I consider this the biggest problem that we've run into, which is the vaccination status source. There are few countries or jurisdictions that have accurate vaccination registries. It's made more difficult by the fragmentation of vaccine delivery across clinics, pharmacies, schools, long-term care facilities, hospitals. There's probably more than I didn't list. Populations move in and out of healthcare systems. So, for example, you'll see we worked with the Kaiser Permanente system a fair bit, but people move in and out of that insurance scheme. The vaccine duration of protection varies. So, for example, 5+ years, I mean, we're not really sure how long they last, but PCVs clearly last for a while, as do other conjugates. On the other extreme are seasonal vaccines like influenza. RSV, we're not even sure how long it lasts, maybe 2 to 3 years. Where data are available, you may not be able to distinguish between vaccines. For example, we've run into problems trying to figure out whether pneumococcal vaccines means a conjugate vaccine or a polysaccharide vaccine. It may not, the data source may not distinguish by the manufacturer. For example, in current times, PCV15 versus PCV20 or Pfizer's versus GSK's RSV vaccine. There are some solutions to this. A national registry is the nicest solutions if you're going to work in an entire country. But like I said, few countries actually have those at a high enough quality for regulatory purposes, or I would even argue for any type of robust study purposes. There are self-contained health systems such as Kaiser Permanente, and those are really nice when they're available. There's the ability, if you have the ability to link multiple data sources, such as hospital and clinic records and incomplete registry, that's really helpful. We did a study in Louisville where we actually went back to the Medicare and Medicaid databases to try and find vaccination status because it was incomplete in patient records. And then tokenization is sort of a new idea or a new term, I guess, for an old idea. And this is a slide about this. I won't go through all of this, but this presentation, I mean, anybody can have this. I'm happy to send it, or if the organizers make these available, that's all fine. So study tokenization transforms identifiable patient information into irreversible random strings of characters that have no meaningful value. And it can be used to perform de-identified analysis of patient data. And these can be, I'll go to that third bullet, tokens be generated using various combinations of available personally identifiable information. And there's some of them listed here. So for example, first name, last name, date of birth, gender, zip code. And on the right side, it just is sort of schematically showing that you can use these tokens that are embedded in a particular database, including, to Steve's question earlier, in clinical trial data that then can be followed through real world databases later on using that token, as long as you can generate the same token based on the same personally identifiable data. And so we've been working on developing this further in some of our studies. So as far as vaccines go, it's not just the vaccine status source, but also the definition of who's been vaccinated or not vaccinated. And even if completely accurate vaccination status is obtained, exposure definition is not obvious. And I'm just going to go through a few examples that we've run across. And this is for adult PCV. So you could be pneumococcal vaccine naive. I mean, you could define it as that. You only want people who have never received a pneumococcal vaccine. So it's this pure population. But in many countries, I would argue in most countries in Europe and the US, this is a really special population that's older adults who have declined not to have any kind of pneumococcal vaccine. It may make the study much longer, even if a representative population could be found, requires robust knowledge of vaccination history for 10 plus years. If you really want to exclude the possibility that any pneumococcal vaccine could have had any effect on the study results. We've looked at no previous PCV so that you could have a polysaccharide vaccine. In some countries and health systems, this is also a special population, also requires robust knowledge of vaccine history for some period of time to make sure that no PCV had been received. You could say no previous PCV or PPSV. You could have no restriction. 20 minus the PCV13 serotypes or PCV20 minus PCV15 serotypes. The problem with this is often regulatory agencies and public health agencies are most concerned with the common serotypes to ensure there's no reduction in the standard of care. The new serotypes, that issue doesn't really exist because that's better than nothing. And I think almost everybody would agree with that. But if you're replacing a vaccine that has that you use a new vaccine with slightly lower immunogenicity to replace an old vaccine, how do you know that that new vaccine is doing the same job for the common serotypes? It also limits which vaccines can be evaluated, and this type of evaluation will become ever more complicated, I would say, when market fragments further. And then for RSV, there are a few permutations, but this is a window of opportunity before RSV vaccines are widely used, and there's lots of different ones to get really good information on how these vaccines work. And currently, it's a single dose. It's unlikely to last more than 2 seasons, at least at high efficacy or high effectiveness. And that limits the need for data history. But there's already some issues. So Pfizer has a maternal indication for its RSV vaccine, and there's also a monoclonal antibody. And having those two together also complicates things. And for sure, if you do an evaluation of vaccine, you're going to need to know whether the person received monoclonal antibody and whether they're a naive population or not. The reverse may not be true. For example, preterm infants in the NICUs for months probably don't have much antibody from a maternal vaccine left. So you might be able to look at monoclonal antibodies even without having knowledge of maternal vaccine. Okay, so the other big area that we've run into is just looking at outcome definitions, and here are just some generic issues. So when you're talking about real-world databases, it often will rely, and at least in the US and Europe, rely on ICD coding, usually ICD-10 these days. That prevents symptom-based definitions, which are often preferred by regulatory agencies. It requires selection of combinations of codes that are often poorly defined and not standardized. You need to determine which coding position. So for example, if you only accept those ICD-9 codes, for example, a code for pneumonia, but it has to be in the first coded position, that may be more specific, but if you put it in any coded position, it may be more sensitive. And I say may because few studies have actually validated ICD coding with chart reviews. So unless you do within your study have an embedded chart review to validate the coding, it can be uncertain whether the outcome you're looking at is really the outcome you want. You may have a poorly defined case definition even with chart reviews, and we've run into this with pneumonia and LRTI case definitions. It's even more problematic when you use non-traditional outcomes. You require long-term follow-up, or you require linking to other databases. And so here are some examples: congestive heart failure, chronic lung disease, and these are appropriate for either an RSV or a pneumococcal vaccine, asthma, antibiotic resistance, long-term care utilization, or other healthcare utilization outcomes. Severity is based on utilization and not symptom, signs, or tests. So, for example, you base it on things like hospitalization, ICU admission, intubation. And again, those are not what's preferred generally by regulatory agencies, which tend to like things like oxygen saturation or specific signs and symptoms indicating a severe outcome. And then there's also difficulty linking with radiology or test outcomes. I put pulse oximetry, but it could also be etiologic outcomes. And so here's just an example of some potential outcomes for post-licensure analysis of PCV vaccine effectiveness. I'm not going to go through this whole table, but you can see there's 4 outcomes I listed: invasive pneumococcal disease, vaccine type otitis media, vaccine type pneumonia or LRTI, and then all-cause pneumonia or LRTI, and then some characteristics across the top. For example, like whether a lab test is required, study designs, outcome definitions, which type of serotypes or not serotypes you might want to look at. This next column is spatiotemporal VE variation. So one criticism that is often leveled against all-cause outcomes is that they can vary based on the occurrence of other conditions like flu or RSV. The VE presumably will go down if more of what you're looking at is flu and RSV. But that's more of a quantitative issue because serotype VE the VE for pneumococcal vaccines differs by serotype, and serotype distribution differs by time and place. So you don't really get rid of that issue, you just have it less if you look at serotype specific outcomes. And then cost and the speed of being able to do these. And IPD is sort of the obvious one, and that's actually already accepted by regulatory agencies, and it's the basis of licensure for PCVs. And regulatory agencies generally in Europe have required us to do post-licensure studies to show that the vaccine still works against IPD. So that's a regulatory commitment in order to maintain the licensure, although the indication is given before the study results are obtained. So that's sort of a given, and what you'd really like are to have those other 3. But vaccine type otitis media is essentially impossible because nobody collects those data. Vaccine type pneumonia or LRTI requires currently Pfizer's serotype specific UAD because that's not really available elsewhere. I mean, there's a few special circumstances like in the UK, and the CDC is evaluating Pfizer's test right now. And then that leaves you with all-cause pneumonia or LRTI. And we've done quite a bit of analysis of these, and this is a few examples I'll just give you. So this was a study we did with Kaiser Permanente in Southern California.
Brad, this is Tom. Sorry to interrupt, but I'm going to have to ask you to cut it a little bit short. We're running short.
Okay, I can run through these really quick because right after this is the summary. So these are just some examples. And you guys are welcome to go through these. And the thing they all have in common is that they were only used for vaccine technical committees. None of them were used for regulatory purposes. And they're all based on all-cause outcomes, not etiologically specific outcomes. And the last one was the CDC, and a group in Germany did some independent ones as well. And then the counterfactual, I'll just mention, is a post-marketing requirement study. This is an observational study. It's not using a real-world evidence database in the sense that it's an administrative database, but it is observational, and it has been accepted as a study that allowed us to have a pneumonia indication in our PCV20 label for the FDA. And I won't go through the design of it, but this is the summary. Real-world evidence and data are already accepted by regulatory agencies in some cases, such as IPD post-licensure commitments. Prospective hospital-based observational data is accepted by regulatory agencies, and that was that I just mentioned. Real-world evidence and real-world data are already accepted by vaccine technical committees, including the use of all-cause outcomes. So those studies that I was showing have been used across Europe and in the US by vaccine technical committees. What remains unclear is the extent to which regulatory agencies will accept those types of study for label enhancement or new indications. And there's a variety of complexities that exist, and some of those could be ameliorated by use of clinically defined or all-cause outcomes, but those also raise some concerns. And I'll stop there. Thank you.
We are running over time, but as the coffee break is quite long, we are going to cannibalize a little bit of the coffee break now. So we have time for 2-3 questions.
Did you have any questions from the people online, Peter? No? Okay, so we'll go back to the room here, ask if there's any questions for Brad.
All right, totally clear or totally confusing?
Totally clear. I guess people feel sorry about your still being awake. So
I'm gonna
Brad, I mean, let me ask you, I mean, you know, the first presentations, as I don't know if you heard, did you hear Helen's and Jacob's presentations? Yeah,
I did.
They were very focused on outbreak situations, right? And what you've been discussing is a little bit the other side of things, you know, very routine situations. How do you see, you know, regards to, let's go back to PCV and RSV. I mean, do you see any of the situations that we discussed related to Ebola, COVID, etc., applicable or useful in the light of, you know, the more routine vaccinations like the ones you work with?
Yeah, I mean, I think that was what I was trying to illustrate a little bit, was that real world evidence is something that's really incorporated into a lot of the thinking already in Europe and the US. So those types of studies are already used routinely, I mean, across almost all the aspects. So they're the basis for safety studies. They're used to support you know, things like the evidence to grade framework with the ACIP. They're used by JCVI. They're used by groups like RKI. So those types of data and databases are already used. I think but the topic of this meeting is to be able to use those types of data for regulatory purposes as well. I mean, to get vaccines licensed. And so I think that that's where we have commonalities with some of the discussion that was given earlier. And that's where the limitation is. You know, can you do these studies and find results and come to common ground with regulatory agencies so that you can have those indications in your label, or that you can guide physicians on whether it's actually working against those types of outcomes? And I think that that's really the big challenge, and that's the one where, at least in our group, we've spent a fair bit of time working.
All right. Yeah, and Bob is on the microphone with a question. Go ahead, Bob.
Bob Chen, Brighton Collaboration. Brad, one potential solution in terms of the vaccine exposure capture is to increase the availability of 2D barcodes at the final delivery point. And I know this has been an issue that historically, when you bring it up with the packaging folks, they kind of resist. I'm just curious, from you, from the EPI side, have you been trying to pressure those folks to kind of do their best to include the availability of 2D barcodes?
That's a great suggestion. All I can say is that I haven't personally, so I don't know if other people have been working on that aspect and to try and get barcodes added into it. You know, I will say that it still doesn't, I mean, unless everybody does that and routinely enters it, it doesn't address some of the issues. Like for COVID vaccines, I think I've had 5 doses at this point, and I received them at 4 different facilities. I mean, I had one, one at an airport, I had one at some makeshift clinic that I took my daughter to, I had another one at a hospital, and I had 2 doses at pharmacies. So all of those groups would have to be coordinated in collecting barcodes and putting them into the same, or at least a database that could be linked in order to overcome this issue.
Yes, but a starting point is to have it on the vaccine vial, and
Oh, yeah.
definitely and the one thing was that the emergency use authorization waived that requirement for all the manufacturers. And I don't know if Pfizer ultimately, once you guys got licensed, whether in fact you did add it in or not. I presume you did, but again, it just highlights that we should try to look for ways of improving that exposure capture information as well.
All right. So thanks again to all the speakers, and I'll hand over to Peter to introduce the next session.
Thank you. I'm asked to introduce Liz Miller, and Liz has also a very extensive bio. And so I asked her, and she asked me to summarize it as a leading scientist in vaccine efficacy and safety, and she works at the London School of Hygiene and Tropical Medicine. She will be our next chair. Thank you.
Thanks, Peter. Well, this session will be looking at how real-world evidence complemented and facilitated the emergency use authorization of COVID-19 vaccines. The emergency use authorization didn't obviate the need for randomized data on efficacy from clinical trials, but the duration of those trials and the size of the safety database could be reduced for the commitment to produce real-world evidence. And again, it's in the context of an emergency need for this vaccine. So to address this session, which will take place both before and after coffee, and I think we need to reduce then the coffee duration. We got 2 very experienced speakers from the US, representing the 2 great institutions in the US in terms of vaccine and biologicals, which is the FDA and CDC. So our first speaker before the coffee break is Hector Izurieta, and he works at FDA. I've served on committees with Hector. He has an enormous experience. If there was any discussion of needing data for efficacy, Hector would always say, "Oh, I've just done a study to look at that." So he has been very heavily involved in COVID-19 evaluations, and he will come and address us for the first session in this, for the first topic in this session.
Hello, my name is Hector Izurieta. I work for the Food and Drug Administration. Next slide. My comments are only mine. They do not obligate FDA in any way, shape, or form. I will try to present the regulatory framework we do have. Most people here probably already know it. Then the emergency declaration for COVID, and then the emergency use authorization examples, the licensure of the first, COVID-19 vaccine, and the emergency use authorization. I'm not as short as you know, anyway, and summarize the results. So what's our regulatory framework? I'm going to bore you. This is FDA job. We always do that. They don't invite us to meetings and such. So for licensure, we have the traditional approval. We have also an option which is called accelerated approval. I will speak about that a little bit more. The animal rule approval, which is not used very much, but transfers, experience in animals to humans. And then we have, for investigational new drugs, we do have special authorizations for unapproved products and also expanded use authorizations. These are limited, but I wanted to mention that we have things that are not only emergency use authorization or accelerated approvals. And then the emergency use authorization, which is one of the main subjects of this talk in response to public health emergencies. Going back to them, for traditional approval, we require substantial evidence in humans. For animal rule, we do require substantial evidence in animals. It's basically the same thing, but we need the data in animals. For accelerated approval, we need a surrogate, which is reasonably likely to promise us that protection. We usually use immunogenicity, but for that, and I will remind everybody of this repeatedly, we need a correlate of protection, and then a confirmatory study. For emergency use authorization, as you know, the standards are going down, right, slowly. We are reasonable to expect protection and safety. And then we have the expanded access programs and investigation and new drugs application, extremely restricted, very special cases. I will not waste too much time on them, but they do exist as options. Now, accelerated approval is something I like to spend 1 minute or so on because it's an important alternative. It does have some requirements. The drug needs to treat a serious condition, and it generally provides a meaningful advantage over available therapy. For accelerated approval, the sponsor does need to demonstrate an effect on a surrogate endpoint reasonably likely to predict that clinical benefit. And we go back to the correlates of protection, which is a problem. If we do not have a correlate of protection, we cannot use immunogenicity as the surrogate, for instance. And the sponsor will need what we define as an adequate and well-controlled confirmatory study that must be conducted post-licensure, but needs to be started, or at least the design be completely agreed upon with FDA beforehand. So this could be a clinical trial, but observational studies have been considered here as well. So I will tell you a little story that you all know about, but from our perspective, it's going to be long, going to be, as I said, boring as we usually are, and that is fine. In February 2020, the Health and Human Secretary, our boss, declared a COVID-19 public health emergency. In March, the Secretary declared that we could use emergency use authorizations during this outbreak. In June 2020, we did publish guidance for industry for emergency use authorizations, basically indicating that in order to authorize these vaccines, FDA must determine that the known and potential benefits outweigh the known and potential risk. It's a little cryptic, right? But that's what we did. And once a manufacturer submits an emergency use authorization request, the agency does evaluate the request and determines whether the relevant criteria are met, taking into account what we would call the totality of the evidence submitted to FDA. And I mean that because I will show you examples in which that's not so thoroughly clear. The data needs to be available to FDA for review and analysis, basically. So what happened during COVID? In December 11, 2020, we authorized a vaccine, the first vaccine, in using what we call emergency use authorization. Interestingly, we did not use emergency, for this emergency, we did not use real-world evidence. We cited the totality of the evidence and issued an emergency use authorization. But we had clinical trial data, randomized placebo-controlled trial data for more than 37,000 individuals, placebo-controlled trial, as I said. The effectiveness data included more than 36,000 people, and unhappily, the available data did not allow a determination of waning of immunity. So we didn't know how long the protection would last, but we had very reasonable data from randomized studies that allowed us to safely authorize that vaccine. Participants did continue to be monitored once the authorization was issued. And in May, we expanded the authorization to a younger age group, 12 to 15 years of age. Now, 8 months later, using another tool, which is what we call the priority review. So in 8 months following the authorization, we did license this same vaccine for individuals 16 years of age and older, citing data on effectiveness that we did review on 20,000 vaccine and 20,000 placebo recipients. At least 12,000 had been followed for 6 months or more. And we did conduct a rigorous evaluation of the post-emergency use authorization safety data, and they determined that myocarditis and pericarditis were serious risks, particularly within the 7 days following the second dose. In fact, during days 0 to 7, so it's 8 days, but we are not very good at counting. The vaccine continued to be available under emergency use authorization for younger age groups and for some immunocompromised individuals. So just to remind you, here we use priority review, but we license a product. And then emergency use authorization for a booster dose. And here we did use real-world evidence, so I will spend a few slides on this. Pfizer submitted data, including real-world evidence, referencing several observational studies suggesting waning of protection for the 2-dose schedule during the Delta variant surge at that time. They also included data on safety and immunogenicity against the Wuhan strain, which was no longer circulating, of course, on approximately 300 adults. Effectiveness was based on immunobridging analysis using correlative protection. So we convened an advisory committee meeting, what we call the VRPAC, to discuss the booster dose. The presentations during this VRPAC meeting, including data from CDC on vaccine effectiveness over time. They also included data from observational studies in Israel on waning of protection and effectiveness of the booster vaccination. CDC did not review that data. The VRPAC committee members voted 16-2 against booster dose approval for ages 16 and older, so against licensure. But asked to vote on emergency use authorization using this available data, the committee did vote 18-0 in favor of the emergency use authorization for 18 years or older. On September 2021, a few days later, based on the HHS emergency declaration and considering the totality of the data available, which included, as I said, real-world evidence, FDA authorized the booster dose for individuals 65 years old and older under emergency use authorization. We also authorized it for 18 through 34 at high risk for severe COVID and for 18 to 64 in both cases for people with frequent institutional exposure, basically healthcare workers. I will very briefly go through one of the presentations from data from Israel. Some of these data has been presented. They presented data on booster protection. They showed waning of immunity observed across all age groups. You can go back to the records that I referenced. And they also showed waning of immunity for severe disease in ages 60 and older for the 2 doses. Then they started a campaign with administration of the booster dose and showed a relative risk for booster vaccination, which was between 5 and 11-fold lower than for individuals who had been boosted using 2 different methods, and concluded that the booster dose was effective and the safety profile. I did not present the safety data, but basically myocarditis was not a problem among that elderly age group, and they considered it was a satisfactory experience. So, in summary, FDA does provide multiple pathways for vaccine access. That includes not only accelerated approval for licensure, but emergency use authorization for an investigational product. Both can allow the use of real-world evidence. Emergency use authorizations provide an expeditious pathway for larger access to investigational products. Other options, as I said, include expanded access programs, investigational new drug applications. During the pandemic, we were able to use real-world data to aid in decision-making for COVID-19 vaccines. The example I presented was on booster dose authorization. And I'd like to acknowledge David Caslow, who is my direct supervisor, so I always recognize him. The 3 of them substantially provided inputs on this presentation, and other individuals, many other individuals at FDA provided the elements for this presentation. Thank you.
Thanks, Hector. Very clear presentation. A nice illustration of actually real-world evidence feeding back to allow licensure, albeit EUA. Approval. Any questions? Peter, is there anyone online with a question? And any? No? Not for the time being. Anyone in the audience? Bob.
Bob Chen Brian Collaboration. Hector, as you know, this was a very challenging time for the agencies, and then the different point, there are different evidences available, and hard decisions had to be made. And at FDA, some of the senior staff at CBER resigned due to some of the decisions that were made. And kind of off the record, are you able to comment in any way, in retrospect, of what kind of obviously these were kind of any lessons for us as public health professionals?
Yeah, I was born in Uruguay, and at a meeting, a friend of mine said to that meeting they were singing and dancing and all that. And somebody says, the Uruguayans should also dance and sing. And he got up and said, we Uruguayans are boring and we dress in gray, in color gray. I will say the same thing. We at FDA do not comment off the record. Thank you.
Any other questions from the audience? Tom?
So again, I don't know if you can comment, but I've been wanting to ask this question, and this is not just for you as well. I mean, looking back, at the pandemic, and of course the outcome of the clinical trials was great, and everybody got very excited. My question is, could we have done without? What would have happened if you would have just said, go ahead, as long as you can manufacture, go ahead and vaccinate, and we'll see what happens? Has that ever been considered?
Yeah, as Bob and you have mentioned, this was a very challenging time. And I asked the question regarding the West Africa outbreak of Ebola, because many thousands of people died. And the outbreak lasted from 2012 to 2015 or 16, depending on who you ask. And there was no vaccine available, although safety data had been already made available. And the disease was extremely deadly. So it was more than 4 years of a very large outbreak. And now in COVID, we could do a little better. We got vaccines approved under emergency use in a few months. It was a record. The question is, can we do better, right? When lives are at stake, where people are dying in large numbers, can we do better? I believe we can. I think this meeting will help us get a little better at that. But I have to admit that we were very successful compared to precedent. That's not enough. When people are dying, my personal opinion, and this is my opinion as a scientist, right? My opinion as a scientist is that at any moment, you have to make a decision, and not making a decision is already a decision, and there are consequences for that decision not to do anything. That's a general statement, doesn't apply to any particular case, but I firmly believe that at any time as a regulator or as a public health person, I'm given the opportunity to make a decision, I don't have the luxury of not making it. I have to use the data available at the time to make that decision. That said, I think we were as successful as we could given the very stressful situation and the very limited restrictions we had in our decision making. I think I'm satisfied with that.
Got a couple more questions before
Yeah,
Steve Black.
Hector, I'll ask you for another off-the-record comment. The U.S. approach used randomized trials, and I'm wondering for future, and one of the consequences of that was that perhaps vaccine confidence was increased in the public, whereas if you just put it out there, the acceptance may have been less. So I'm wondering for a future pandemic in a country with as much divisiveness about information as there is now, whether it would even be advisable for a disease with relatively low mortality to consider going straight to introduction rather than going through the time delay for a randomized trial. Over.
Yeah, it's a very good question because this is one of the dilemmas we have in the US. As you know, you mentioned it, we have a very complicated system with very extreme opinions sometimes that can drive the decisions regarding supporting vaccination in one direction or the other. So unhappily, that is part of the reality. That's not data that is submitted to regulatory authorities. It's just the environment in which we live. So I think that considering that, we did the best we could by authorizing the vaccine under emergency use using strong clinical trial data. But that's just my opinion. But as you very well said, it's a complicated situation.
One final question, if you'd just introduce yourself.
Hello, everyone. This is Ahmed from Saudi Food and Drug Regulatory Agency. My question to Hector is, in regards to accountability and responsibilities of assessors versus committee members, how do they interact with each other? If there's who is responsible for making the decision, who is responsible for collecting the data? Do the committee also participate in collecting information and reviewing data as well? So this is the question, and also how do the CDC interact with the US FDA? Which one determines that is an emergency situation, or this is a regular pathway for a vaccine, etc.? Thank you.
Thank you. I think you have asked extremely pertinent questions, all of them relevant. So I will try to address the first one first. We had a very strong relationship with CDC. In fact, I saw Tom Shimbabukuro here, and I say hi to him. That's evidence of good relationships. Welcome, Tom. how do we work with advisory groups? We call our group the VERPAC, right? We at the FDA are responsible for the decisions. We do select the advisory committee people. We want them to be independent of us. We provide them information regarding the data we obtain from the sponsors, so they have sufficient information. They do not review directly the data. We are responsible for that review, and we use their advice as that. It's their advice. We are not compelled to follow their advice. In some cases, we haven't. I have presented examples in which we have not exactly followed it as advice. We prefer to follow the advice because it also helps with a number of circumstances, including the environment in which we live. But we are the ones responsible for the final decision. It's not the committee. And we do consult with CDC, but not in regard to whether we license a product or not. We do consult in regard to recommendations. Sometimes we discuss, the options for labeling. They are extremely well informed. They have huge public health capabilities. In fact, I worked for 11 years with CDC as well. So we have a strong respect for CDC. We count on them whenever it's appropriate, and we always communicate with them. But we can discuss offline afterwards.